small electric currents in intravascular thrombosis. Produced with the assistance of the National Heart Institute, the National Institutes of Health, Bethesda, Maryland. Produced by Dr. Philip N. Sawyer, Department of Surgery, State University of New York, Downstate Medical Center. And Dr. Roman Vishniac, Graduate School of Science, Yeshiva University, New York, New York. With the assistance of Dr. E. Suckling and Dr. S. A. Wesolowski, State University of New York, Downstate Medical Center. Electrical potential differences exist across the membranes of virtually all living cells. They are a measure of activity within the cell and are a manifestation of cellular membrane control of ion transport. On the basis of this mechanism, many living organisms, such as the electric eel, have developed very high voltage batteries which they use as weapons. Past experiments indicated that spontaneous and applied currents will produce occlusion of large vessels. The authors investigated the effect of small applied electric currents on blood flow through vessels seen through the microscope in order to measure the visible reaction of the vessels, the contained blood cells, and plasma to the current. The question was posed, is it possible to visualize occlusion taking place in blood vessels of the rat mesentery and mesoappendix when an applied electric current is passed across them? The following setup is used. A short midline incision is made in the abdomen of an anesthetized rat. The rat is attached to the manipulating board made of lucite. This is attached to a micro-manipulator. The cecum, with its attached mesoappendix, is extracted through the incision. The blood vessels of the mesoappendix are covered with a thin endothelial layer. When transilluminated, they are clearly visible and make excellent test vessels. The entire experimental animal can be moved on the stage so that the desired vessels of the mesoappendix are visualized. To the left is a flask of 1% gelatin Ringer's solution used to bathe the mesentery. Dr. Peter Suckling is inserting the first microelectrode into the manipulator. This is one of a pair of microelectrodes which is used to pass current across the mesentery. He next receives from Dr. Wesolowski a microammeter, a set of batteries, and a decade box, variable resistance which will be used to control the total amount of current passed across the mesentery by the batteries. On the other side, Dr. Wesolowski is inserting the second electrode into its manipulator, which he has just placed on the table. The electrode manipulators are located on either side of the mesoappendix. Dr. Vishniac is putting together the cinemicroscopic apparatus to photograph the clotting process in the mesoappendix. This technique was used to determine whether small applied currents could produce thrombosis in the mesentery. The mesentery is centered over the transilluminated lucite rod. The electrodes are placed on either side of the mesentery. The negative electrode on one side, the positive electrode on the other side. Dr. Vishniac is examining the mesoappendix through the beam splitter of the cinemicroscopic apparatus, preparatory to starting photography. The selected scene is easily found under low power, photographed, then viewed under increasingly higher power until the desired magnification is attained. The scene shows normal flow through several small arterioles and a large vein in the center of the picture. It has branches on either side. A better view under higher power shows rapid flow through several vessels. Current flow has started across the mesentery. After a few moments, white blood cells begin to stick to the blood vessel walls. They roll down the sides of the walls like little hoops. The electric current is causing thrombus formation close to the branching of this large vein. This thrombus is formed by close approximation of the current passing electrodes. It grows rapidly in size. The vessel is entirely occluded in approximately 10 minutes by a current of 25 microamperes. The flow in most of the small vessels in this scene has completely stopped. The electric current causes rouleau formation in all of the blood vessels. Here, current of 40 microamperes has been passing for 5 to 10 minutes across this mesoappendix. All of the vessels are filled with clumps of cells interspersed with large refractile bodies, presumably fibrin. Gradually, the flow stops and ultimately comes to a complete halt. As pressures increase in other areas of the system, one or another vessel will occasionally open up and extrude its contained thrombus. This is rouleau formation in a vein showing rapid flow. White cells and red cells can be seen attached to the vessel walls. Note the considerable diapedesis, or migration of red blood cells through the vessels into the surrounding tissues. Again, with the passage of time, flow progressively decreases. The capillaries occlude first, followed by occlusion of large vessels seen in this picture. The cells in the right-hand branch of this Y form clumps and rouleaux, stuck together by some kind of protein, probably fibrin. As a further step in occlusion, the direction of flow reverses. It is now going in the opposite direction down the two right-hand branches of the Y. All of the small branches have occluded. Further progression of rouleau formation takes place as long as current flows. Probably this process takes place because of the change in the ionic atmosphere of the blood flowing in the blood vessel exposed to the electric field. The change is caused by current flow. A correlated hypothesis suggests that the electric current is changing the blood sol in the blood vessel to a gel by depolymerizing the fibrinogen and forming fibrin. The blood cells are caught in the solid fibrin coagulum. Now all blood flow has virtually stopped. There are no evidences of flow in the small capillaries and arteries. Progressive hyalinization is taking place in the vessels, a further stage in thrombosis. The lower vessels in this venous capillary communication are occluded. Thrombosis progresses rapidly under the influence of the electric current until occlusion is complete. The last few moments of flow display good examples of cell clumping and formation of refractile emboli. The next scene shows high flow, no flow. Blood continues to flow through the lower branches of this three-cornered communication, but not through the upper branches. The upper extension of the vessel is completely occluded by a current flow of 20 microamperes for 30 minutes. Separation of cells and plasma is visible at the point of junction between the thrombus and the flowing blood. Here is a completely occluded vessel, the result of 10 microamperes current for 25 minutes. In this view, the entire sequence of small vessel thrombosis takes place. First, disturbance in flow in the right upper branch of the division takes place. Then the walls of all the vessels appear to thicken and finally progressively occlude. Most of these pictures were taken with the electrodes on either edge of the membrane. Occasionally, one was taken with the electrodes closely juxtaposed to a vessel to obtain a specific effect. Occlusions seen in these scenes were all caused by currents smaller than 100 microamperes. The question arises, can living injured tissues cause the same phenomena in adjacent tissues due to the injury currents produced? The results from the experiments carried out to answer this question will be discussed in the next project. Small electric currents in intravascular thrombosis. Produced by Dr. Philip N. Sawyer, Department of Surgery, State University of New York, Downstate Medical Center, and Dr. Roman Vishniac, Graduate School of Science, Yeshiva University, New York, New York, with the assistance of Dr. E. E. Suckling and Dr. S. A. Wesolowski, State University of New York, Downstate Medical Center.